Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Before we jump into this episode, I'd like to share a few things with you. The December and last Dr. GPCR newsletter of 2020 is now available. If you haven't subscribed yet, please visit drgpcr.com/newsletter. Also, don't forget to share the newsletter with your colleagues. We cannot wait to bring you brand new Dr. GPCR newsletter editions in 2021. Second, we will be taking a short break of the Dr. GPCR podcast in January 2021 as our family will be welcoming a baby boy. But rest assured, we are already working on bringing you brand new episodes, starting sometimes in February 2021 with an amazing lineup of guests. Also, stay tuned for a major announcement regarding the podcast. We're also proud to announce that we are pursuing consulting opportunities in the GPCR field. For help with your R&D project, please visit drgpcr.com/consulting or reach out by email at hello@drgpcr.com. And last but not least, we will be closing this first season of the Dr. GPCR podcast with a series of interviews with phenomenal female scientists in the field. We hope you'll enjoy it. Hello, everyone. This is Yamina for another episode of the Dr. GPCR podcast. Today, I have the pleasure of talking with Dr. Debbie Hay. She's coming to us from New Zealand, so she's talking to us from the future. Hi, Debbie.
Hi, really nice to talk to you today.
That's amazing. So you're 16 hours ahead of us. How does your Wednesday look like so far?
It's looking good, actually. It's winter here in New Zealand. So, of course, also, seasonally, that's different to a lot of you out there listening at this time of year. But it's been unseasonably warm and the sun is shining. So we shall see. Maybe it'll be snow at the weekend. Who knows?
It's a great time to talk about GPCRs.
It's always a great time to talk about GPCRs.
Absolutely. Thanks so much for being here today.
No problem. Really happy to have this discussion.
Awesome. So I wanted to talk to you to get to know you better, get to, showcase your research. And I wanted to ask you a couple of questions and hopefully get more people interested in working with you, but also getting to know you as a scientist better. So can you tell us a little bit more about your trajectory as a scientist?
Yeah, absolutely. So I'll start off with what I wanted to do before I'd heard of a GPCR, and I always wanted to be a vet. I have an absolute passion for animals. I grew up with a lot of rescue dogs, and all I ever wanted to do was be a vet. However, I didn't get the grades to be a vet, so I needed to look for something different. And when I was looking and browsing the different courses, I saw pharmacology and thought, hey, that looks cool. Who doesn't want to know how drugs work? So I started an undergraduate degree in pharmacology at the University of Sheffield in the UK. I'm British originally. And that degree gave me an opportunity during the degree to do an industrial research placement with Glaxo Wellcome in Stevenage in the UK. So I worked for a year during my undergraduate degree in the receptor pharmacology unit there, which was really amazing. It opened my eyes to industry, the pharmaceutical industry. I learned so much from that year. But I had to go back and finish my degree, which I did. And then I did a PhD in molecular pharmacology at Imperial College London. And then I met a Kiwi and moved to New Zealand, where I did some postdoctoral research. So I moved to the University of Auckland, the School of Biological Sciences, and I was actually there for 17 years. And I literally just moved from that school. So while I was there, I worked doing postdoctoral research, setting up my own lab, and as a senior scientist for a small biotechnology company. Called Proteimics, and that was another really interesting experience, going from big pharma to small biotech, where I was actually able to visit the FDA and present in a pre-IND meeting at the FDA, and that's not an opportunity that many pharmacologists get. So that was a really awesome thing. And I set up my lab there. I eventually became head of department for a year, a large school. So I got sort of senior management experience as well. But then a new opportunity came up down here at the University of Otago in the pharmacology department. And so I've recently joined there as a professor. It's really awesome for me to be actually in a pharmacology department, having done undergraduate pharmacology and never having actually worked in a pharmacology department. I'm really excited about this new opportunity. So
Going
that's sort of my career trajectory, I guess.
going back to the roots, to the beginning of the pharmacology story. So
Yeah.
you have an amazing set of different experiences. Can you comment a little bit more about, the differences between academia, big pharma and biotech, and what did you learn in those experiences?
I learned a great deal about drug discovery, that's for sure. I mean, running my own academic lab, thinking about potential targets, reflecting on the drug screening that I did in industry, reflecting on the regulatory aspects that I learned about in biotech. I think that's all threaded together really well to certainly help me think quite deeply about what actually makes a good target. But each of those has offered me really interesting experiences, and certainly I would recommend all of those to people out there thinking about careers. They all allow you to do really interesting science. Absolutely, in academia you have, generally speaking, that freedom to do what you like, but of course you've got to get it funded. In the pharmaceutical industry, you may not be able to work on your personal favorite topic, but it's a different experience, and they are all enriching in different ways. And my career trajectory shows that you can dabble in each of them at different times. You don't have to feel that you're stuck in one or the other. And other aspects that I didn't talk about are, I've done paid scientific writing, editing as well. I do consulting from companies. There are a large range of different flavors that you can bring to your career in science.
That's amazing. And we'll get back to this question at the end of our discussion, but so you spent a year working at Glaxo. Was that your first interaction with GPCRs, or when did you first get
Yeah, it was. I mean, I guess I'd sort of heard of them at the beginning of my undergraduate degree, but they didn't really mean that much to me. But when I was joining Glaxo in that receptor pharmacology unit, and as an industrial trainee, you are doing what the other scientists there are doing. You're not an intern making cups of tea. You are actually there doing the screening, so you're getting real life experience. And so the particular program I worked on was a beta 2 adrenergic receptor agonist, long-acting agonist program for asthma. And I was doing some screening for that. So, I mean, that taught me a lot about GPCRs. And there was a real emphasis on good experimental design, analysis of data. And that gave me some really deep insights into GPCRs from midway through my degree. So that did get me started with GPCRs for sure. And then when I went back to university and I did my honours, that was also on a GPCR, because I'd sort of learned about them. They were interesting and I could see their relevance as targets. And that was melanocortin receptors that I worked on then. And I've stuck with GPCRs ever since, as I'm sure anyone who knows my publications will have seen. So actually, the molecular pharmacology undergraduate paper that I was doing, that really inspired me to continue to work on GPCRs, because I learned about these newly discovered GPCR accessory proteins called RAMPs, receptor activity modifying proteins, in that undergraduate degree. And I thought, oh, they sound really interesting. And there was a fantastic new Nature paper that had come out of Glaxo, just down the corridor from where I had worked. So that work was going on when I was there, and I didn't even realize it. And it was a time when GPCR dimers were the thing. You know, right now it's bias. Back then it was dimers. And that was really exciting at the time, and it still is for some receptors, though perhaps not all. And so I saw a PhD position come up at Imperial on ramps, and I thought, right, that's it. I'm going to apply for that. And I've worked on those proteins ever since. It was a really new area at the time, and, you know, potentially high risk, but there was also a lot of scope to make impact.
Agreed. And we're still, I think there is still some work to be done on ramps and understanding in depth their function when it comes to various GPCRs. So one of my questions that I ask everyone is, what is your favorite GPCR? I guess you do have an answer for that, but my question is to you, which one is your favorite ramp, if you have one?
Oh, no favorite ramp. And I have to say, I really, yeah, look, I have a favorite GPCR family. I can sort of subdivide that a little bit, but it's Class B GPCRs, which are absolutely the thing that I'm most passionate about. And the reason for that is because it's a small family of only 15 genes. But those 15 genes create massive diversity because of their association with the ramps, and because there are several splice variants of those receptors. And that's something that we often forget about. Splice variants, they might be really important, and we're just not studying them very much at all. So they're fascinating because just a few genes create actually a lot more pharmacological diversity than you might think. And although they're peptide receptors, and traditionally people think, oh, well, they're not so great as drug targets, that's not true at all in this case. A really high percentage of the receptors in this family have FDA-approved drugs.
Yep.
So they're really therapeutically relevant. Now, of course, within that, my favorites and focus are two GPCRs, the calcitonin-like receptor and the calcitonin receptor, interacting with those three ramps. And so I can't pick a favorite because I don't actually know which is most important. And that's one of the things when we come to perhaps one of your questions later about, you know, the challenges. I can't pick a favorite here because these are such complex receptors, and we fundamentally don't understand which are most important because we don't have selective ligands. We have a lot of cross-reactivity between the endogenous agonists and the receptors, and the antibodies out there to look at them, as with many GPCRs, are not worth the money, shall we say, politely.
Agreed. And so you have two favorite Class B GPCRs. What is the current status research on calcitonin receptors? What do we know about these receptors?
I mean, the biggest thing right now for these receptors is that we've had a large number of drug approvals against the CGRP aspect of this family. So that's calcitonin gene-related peptide, a neuropeptide found in sensory nerves, involved in migraine. And there are now a number of monoclonal antibodies and small molecule antagonists against that target that have been approved in the last couple of years. Mm-hmm. So it's really exciting times for that in particular. So in fact, there is one monoclonal antibody called ireneumab, and that targets the CLR-RAMP1 complex, and it was the first approved antibody against a GPCR. So that was really exciting and first in class for that target. Then there have been multiple monoclonal antibodies against the CGRP peptide ligand approved, and also small molecule antagonists against that canonical CGRP receptor. So really exciting times for that.
So, but physiologically, if I remember correctly, the antibody, it was the Amgen antibody.
That's correct. It's an Amgen Novartis collaboration that's ended up with that drug. Yep.
for the migraine. I read the paper and I thought it was really neat. It was a neat work, and it was exciting to finally see an antibody approved that targeting a GPCR. Um, uh, Pathophysiologically, what do these receptors do? Why would we want to target them?
Well, I don't actually know. I mean, we've got. I say that loosely. I mean, of course, you can read papers and make up a story, but actually, fundamentally, I still don't think we really know. We don't exactly know how CGRP is involved in migraine, and fundamentally, we oversimplify the receptor system in a quite a problematic way. So we all assume that it's that CLR-RAM1 complex that mediates all the actions of CGRP, and everyone says that's what all the drugs target. But in reality, those drugs are not as selective as they say. And there is another candidate, which is another RAMP1 complex to GPCR with the calcitonin receptor, and that's also a high affinity CGRP receptor. And it's largely been ignored as potentially mediating some of that pathophysiology. So people have a bias. They follow that lead in the canonical receptor, but they ignore the pharmacology. And the pharmacology says, actually, no, that ligand binds to other complexes. So I'd love to say we really knew what was going on. I think we can say that CGRP itself, it's elevated in migraine. If you infuse CGRP into a migrainer, they will get migraine-like headache, whereas someone who does not suffer from migraine will only get a mild headache. So it seems that people with migraine are sensitized to CGRP. So clearly there is something really fundamental going on with that CGRP system in migraineurs. But, you look at how the drugs are working. They don't work for everyone. So there's obviously more to it. And for some people who are chronic migraine sufferers, that's 15 or more headache days per month, their migraine's been abolished.
Wow.
So these can be life-changing. But why is there that diversity in response? I think there is so much to know. And really interestingly, for migraine being such a prevalent condition, affecting up to 20% of women worldwide and approximately 10% of men, it's hugely understudied. I go to the big major international headache meetings and they have like maybe 1,000 attendees. Wow. You go to a diabetes meeting, it's 20,000. I think there is so much scope for original research in understanding headache disorders. And migraine is so fascinating because it's not just the head pain, it's sensory disturbances as well. It's sound, it's smell, it's light. How are all those pathways affected for this one condition? What is going on in the brain? What's going on in the peripheral nervous system that creates these amazing symptoms, which for some inspires huge creativity and for others is a disability.
It is. It's debilitating when you're suffering from migraine. I had a colleague who oftentimes suffered from migraine, and those days we wouldn't see this person in the office because it was just a horrible day, and you couldn't.
It's an invisible disability that is so often ignored and should not be. And I think a really important place that scientists can contribute is by focusing some research in this space. We have some treatments, there's so much more we can do, and there's so much we don't understand about this neurological condition.
On the molecular pharmacology level of the CGRP receptor, do we know if it contributes to other symptoms, or what symptoms does it contribute to in migraine?
It has the potential to contribute to a large number. At the moment, all of the trials have principally focused on primary endpoints being the pain. Mm-hmm. I haven't delved into enough detail around whether this improves aura, but the fact that it can abolish the migraine I think is really important. It's not like a triptan that you're taking when you feel the migraine coming on and it stops at that point. This is a preventative, so it's clearly able to block part of the whole pathway somehow that we don't understand, that is then preventing that migraine, which is, the first preventative treatment that actually has been designed to target this condition. All the other preventatives have come from other conditions. They're things like anti-epileptics, and they have side effects that are not very tolerable. So I think it's still early days, and there's an awful lot of scope here. When you get those first drug approvals, they're certainly not the last.
Hopefully not, and hopefully we'll get to better understand the molecular details of these receptor functions in disease settings like migraine, and hopefully we'll get to target them at multiple levels. Who knows with what compounds or what antibodies.
Yeah, but one of the biggest challenges is these are obligatory dimers, these receptors.
Mm-hmm.
And we really need to know exactly where that functional dimer is expressed. So if we don't know where that receptor complex is found, we can't understand the pathophysiology and we can't target it properly. And this is just such a big challenge in the GPCR field, or any membrane proteins really that are dimeric. You can do any mRNA study that you like, but it fundamentally won't tell you what the functional complex is at the cell surface, even if you do single cell. So this is a big challenge in the GPCR space, and it's something that we are feeling acutely with this receptor family, and anyone who works on dimers or ramps has to think about what that complex is in the cell of interest. And there are so many studies that draw together very loose threads and draw conclusions that just aren't good enough for us to really know. As a receptor pharmacology, understanding your target is critical. And I don't think we know enough about these targets at all. Because you have to take into account how each ramp interacts with other GPCRs and how each GPCR interacts with the 3 ramps.
Exactly, and having the correct tools and the correct systems to study these is also a challenge. I remember Harriet, who was on your team back in New York when I was in Palm's lab, where the struggle was to find a cell that didn't have any ramps, so that you can actually study what was the effect of these ramp or each of these 3 ramps on the receptor function. And that's something that
absolutely
difficult. What I wanted also to add is that yes, you need the right system, and you need to figure out where is that dimer expressed in a relevant system. But also an educated guess would be that maybe these dimers are differentially expressed in, for example, patients who have chronic migraines versus healthy people. So that's also
absolutely. I mean, this is it, and it's something about what are the key challenges and key questions still remaining out there in GPCRs in general, and here it's which receptor where in a disease condition and which pathway. We need to know all of those really to be truly successful, as challenging as that may be.
Yes. So we were talking about, the different tools, and you had mentioned antibodies. It's very difficult to find a good antibody to specifically target a GPCR, just only to start with research purposes. What are some of the tools and initiatives you think would be needed to better understand class B receptor function, dimerization in general, and their interaction with ramps in the context of just quote unquote studying the molecular pharmacology, but at the same time trying to tie it to a disease state?
Oh, yeah. I mean, look, I'd love to know the answer to that question because, we do default still to antibodies. I think the arenimab is a great example of something that can be achieved in terms of generating a single antibody tool against 2 receptor components. But I would make the argument that it is not nearly as selective as one would have us believe, in that I have not seen the data for its ability to block or not block the closely related CTR ramp1 complex. And structural data, given the shared amino acids between the receptors and the known binding of arenomab, very strongly says to me that it can bind to the CTR RAM1 complex. So even there, where we have an antibody against a dimer, because we have a shared component with other receptors, that's still not good enough. I'm sure there are some really elegant ways of tackling this, but I don't know really what the best way is. We're looking at doing bimolecular fluorescence complementation in vivo as possibly a way to look at this, where you tag half of fluorescent protein onto one subunit and half onto another. That's been used quite successfully in vitro. There is a little bit of in vivo data, but perhaps this is a way to help us understand, if we make mice expressing these, where a dimer is expressed. But will that work? Only time will tell. Selective ligands are always incredibly valuable, and I come at that always as a pharmacologist, that if we have selective pharmacological tools with all the structures that are coming now, I think we have much more of a chance, but I've yet to see really great success.
And there's been a boom recently of structures everywhere, and I don't want to say they're easy to get. It's not, I don't want to say it's easy to get a structure. I've never worked on getting a structure, but by the sheer amount of papers that come out for all of these GPCRs and show structural data, I think we're getting better and better at acquiring this information, and that might definitely be helpful in better targeting these receptor complexes in specific diseases. If we know at least where the two proteins interact and where the ligand might bind, I think that's something, definitely a tool that can be useful in the future when it comes to better understanding the function of these receptors.
Yeah, it's incredibly helpful. And to now have, from when I started looking at ramps all those years ago, having no information at all, to in the last year or two having one of the sets, CLR with ramp 1, ramp 2, ramp 3, and different ligands out of the Sexton-Wooten group at Monash, plus some of the lovely, really detailed work that Augie Piosak at Oklahoma has been doing. We are really getting a lovely molecular picture of how those ligands are engaging their receptors, and the surprises, the real surprises with the fact that actually the ramp is not providing many molecular contacts at all to the ligand, and is probably principally acting in an allosteric manner to augment the GPCR binding site, which means that making those selective ligands is perhaps quite challenging, because you are driven by subtle changes in conformation between each receptor complex, which is quite hard to then model and get at.
Exactly. And when you get a crystal structure, you get one snapshot, or with a specific ligand and complex, of what happens, but these molecules are dynamic. I think at least I do tend to forget that these molecules are extremely dynamic, and they're not, it's not a one picture that doesn't move. It's always moving, and you have all these components intracellularly, and also the extracellular ligands that are there that modulate the function of these receptors.
Absolutely. As I think Terry Kanakin once said, or says frequently, I don't want to misquote him, but he talks about an energy landscape of conformations. These are in a fluid membrane. They are incredibly dynamic, and that's what makes them so fascinating and able to interact with so many different ligands and intracellular partners. But we do focus on that beautiful one snapshot image, but we mustn't forget that that's just part of the picture.
It is part of the picture, and I think that's on one hand the challenge, but on the other hand That's why people are so interested in working on GPCRs, and that's why we're here today, because it's so interesting and so intriguing on how can you get, for example, you mentioned that class B GPCRs, there's 15 receptors in the class, but there still do so many, they control so many important physiological functions, and there is so much research and also an interest in generating drugs targeting these 15 class B GPCRs. That's why it's still fascinating. I know the answer to this already. So one of my questions that I ask everyone is, do you think GPCRs are still good drug targets? What are your thoughts?
No, terrible. Absolutely not.
We should all we should all go home and bake bread.
Yeah, no, that's it. We're done here. It's all done. No, of course they are. They still are now, as much as they were before. They have been for a long time, even before we knew what they were. And reflecting on Paul Insel's podcast and where things were when he was starting to work on these, they're just as relevant now. They will continue to be. They do take a huge market share. There are so many we don't understand. All we can do is be smarter at targeting them.
And building more tools and more ways of understanding their physiological roles, and hopefully their role in pathophysiology as well. What do you think is the key information that's missing from a general point of view GPCRs, but maybe class B GPCRs, especially your two favorites, to speed up drug discovery targeting these receptors, but also understand their biology?
Yeah, I mean, I think I sort of hinted at this before, but I'll reiterate this. I mean, we have to know where our receptor is in our tissue of interest, in our disease condition, and how it is signaling, because that is what we need to target. Whether we're looking at an agonist to promote a beneficial effect or an antagonist to block something that we don't like, we have to truly know where our target is and how it's functioning in a disease relevant condition. I have used model systems, for the majority of my career, and they are still incredibly valuable. But if we can get into human tissues, if we can use, more of those sorts of models, and we look at how the NIH is encouraging target validation and multi-species validation, the use of different labs, different tissues, different models coming together collaboratively to have real confidence in our data. We must do things with good experimental design, with sufficient replication, with appropriate statistics. Often in my lab, if we see something interesting, I'll get someone else independently to replicate that in my own lab, or sometimes in someone else's lab, because you need to be sure about it. And unfortunately, there is too much out there that is not replicable. And it causes careers to die. It's just not fair on the junior scientists who then follow up on that work to find it can't be repeated. So I'm pretty passionate about robust science as well.
That's the way to go. I mean, without that robust science and that collaborative, mentality, it's very hard to move ahead, because I think there's so much work to be done in the field from so many angles, including having model systems, but also having crystal structures, and also having tool compounds, and having the assays to understand the pharmacology of receptors, that we need to be working together. And definitely we need solid reproducible data that will get us closer to those answers and hopefully speed up GPCR drug discovery.
Yep, absolutely.
So you've mentioned in the beginning, and we've touched about it right after you told us about your stints in industry, pharma, biotech, consulting, also editorial work, and being in academia. What would be your advice to junior scientists who are interested in GPCRs but don't necessarily know which way to go in their career?
You have to have a burning curiosity. If you don't have a burning curiosity that fuels your desire to do science in whatever way throughout your career, then give it up, is what I would say. It's hard. Science is hard. All of these careers are hard. And some days you'll go home and you're fed up and you didn't get that grant and you don't want to do it. But if you've got that deep, deep drive still to get over those failures and pick yourself up, and that's a strong enough fire burning, then you will succeed. If you're like, oh, it's kind of interesting, but I'm not really that passionate about it, then maybe you just want to do something else. It's really, really hard, and I've seen many strive to succeed and fail. Perhaps they reflect in time that their heart wasn't in it. I think it's very hard to know, and I think it's very easy to be led by what others are doing and forget what it is that really matters to you. I'd also say that you must recognize and celebrate the successes and wins when they're there, because they will be outweighed by the failures. Be they the experiment that didn't work, the paper that gets rejected for the 7th time, be they the fellowship you didn't get, the grant you didn't get. We have to allow ourselves, and I allow myself one day of being grumpy about it, and then I move on, because you can't focus if you're unhappy with that. But you must recognize and celebrate those wins, however small they are. They're really important, and we must enjoy those moments.
How do you celebrate the wins?
Usually. It depends. We'll have lab afternoon teas, morning teas. We might go out for a few Friday evening drinks or something like that. I think even just recognizing it in small ways, saying thank you, acknowledging those who have helped and supported. It can be done in lots of different ways.
Especially now with this whole COVID situation, it's very hard to get together. So I think, picking up the phone or setting up a Zoom call and saying, hey, good job, or thank you for helping, or thank you for putting in the work, can definitely change the person's perspective and, make a difference in their day.
And we can all do it better. Yes. I know I don't do it enough. I know I should. So yeah, let's all remember. And our Prime Minister here talks a lot about just being kind. Be kind to others. And I think that's a message that's really important.
And I think there is no other better way to end our discussion than this. Be kind, say thank you. And you don't need to buy a yacht to the person, for the person. Just say hi and say thank you whenever you can.
Yeah, absolutely.
Thank you so much for your time, Debbie.
No problem. Great to talk to you today. Thanks a lot.
Thank you. Bye-bye.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Subscribe to the podcast wherever you listen to podcasts. I'd like to thank our guest, Attila Forrest, Jin Chong, and Shivani Sajdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. We're always excited to hear from you. Visit us at drgpcr.com or send us a note at hello@drgpcr.com. I also wanted to wish you happy holidays and a very happy new year. I'd like to thank this year's guests and thank you for being such a wonderful audience. Thank you again for the privilege of your time, and until next time, stay safe.